Hi everyone, it's Mr. Sinti, and today we're going to talk about one of the classics of biology, which is called transcription. It's how DNA makes RNA, and it's the first of two steps in protein synthesis. The first thing that you have to do is make RNA, and then RNA is going to be an intermediate molecule, and it's going to be transcribed in the nucleus in a eukaryotic cell. And so transcription, very simply, is, if you think of the word itself, it's a word that means to transcribe or to transfer by writing. And so you might think of the old days before a Xerox machine or even before a printing press. There used to be individuals that if you wrote a book, you'd have to literally scribe it. A scriber would take it and copy it into another book. And so it's a pretty tedious process, but in the cell, we have an enzyme that will speed up this process of turning DNA into RNA, so it's rather quick. And so transcription is the making of RNA. And as it turns out, there's going to be four types. There's more than four types, but we're going to talk about four types of RNA, and in particular, three of them are going to be really emphasized. But all of them are produced the same way, which means DNA opens up and RNA is produced just like this. Remember from the last lesson, RNA is a single-stranded polynucleotide, and it's very similar to DNA, except it has the sugar ribose and it has a new nitrogen base called uracil. And so let's just jump right into this process. And so let's get into this and say, let's go down here and say that transcription, I'd like to say transcription occurs in the nucleus only. Because that's where DNA is, and I can say RNA is made in the nucleus, but the truth is that that's only for eukaryotic cells. For prokaryotic cells like bacteria, transcription occurs right in the cytoplasm. And so right out of the gate, here's a picture of a eukaryotic cell, like for example us, and we have this nucleus, and DNA is really big. These highlighted areas like yellow, red, brown, and green, these areas right here are segments of DNA, or we'll call genes. In other words, this is the recipe to make RNA. This is the recipe to make RNA. This is the recipe to make RNA. And these are the different kinds. The one that I'm going to really emphasize today is messenger RNA. And the reason it's called messenger RNA, let me make that a little bit smaller here. The reason it's called messenger RNA is because it might be obvious that it carries the message of the genetic code from the nucleus. So in other words, here's the RNA that's being produced. It's going to travel out into the cytoplasm, and it contains the message of how to make a protein. And so it's little m RNA. And then there's other genes, not the same one, but there's other genes that stand for tRNA, and T stands for transfer. Or sometimes I like to call it taxi RNA, but it's really called transfer, or little t RNA. And that's a molecule that's going to carry amino acids to the ribosome. And that's what we're going to look at when we look at how messenger RNA is decoded and how transfer RNA brings amino acids to the ribosome. And speaking of ribosome, is rRNA, or ribosomal RNA, or rRNA. Kind of corny, but this is the favorite type of RNA for pirates because it's R rRNA. And it's actually, there's only one gene shown here in this picture, but actually our DNA contains many, many genes of RNA, which is kind of curious. They're kind of repetitive, and they're often next to each other in tandem. And we believe that it's because there's just a tremendous amount of rRNA needed, because rRNA makes up a ribosome, if you remember that. Like for example, let me just say it while we're here. A ribosome itself is made up of proteins in two subunits, a small and a large, and there's rRNA wrapped around it like this. And that's what we think a ribosome is composed of. Transfer RNA kind of looks funky town like this, and it carries an amino acid with it, like that. And messenger RNA looks just like this, kind of linear. a straight line with it. These are the nitrogen bases sticking out. Here are the nitrogen bases. I didn't draw any nitrogen bases, but now I am. And then what's this all about? snRNA, which stands for small nuclear RNA, or snRNA. There's a few functions of small nuclear RNA, but one in particular, and I'll talk about it actually in today's lesson, is that it actually helps to edit the messenger RNA when it is transcribed, and so it shortens the messenger RNA. So we'll talk a little bit about that. So these are 3 types or 4 types of RNA if you want to include small nuclear: messenger RNA, transfer RNA, ribosomal RNA, and small nuclear RNA. Sounds intriguing, doesn't it? So to get from DNA to the language of a protein, which is a sequence of amino acids, which is a polypeptide. So here's where the ribosomes are. If this is a ribosome out here in the cytoplasm, here's another ribosome, not drawn to scale. But this is where proteins are made out here. So DNA is inside the nucleus. It has to make RNA, and that RNA leaves the nucleus, and transfer RNA leaves the nucleus. And so what we say is that to make a protein, it's broken up into these 2 classic, and I use the word classic probably too much, but this really is an example of where somebody would use the word classic, not just me. But classic meaning that, I'm going red here, protein synthesis is made up of 2 major processes, which is transcription and, secondly, translation. And so transcription is taking your DNA and transcribing it into RNA, and that's what we're going to look at in this podcast. And then in a later conversation, we'll talk about how the RNA is translated. In other words, when you use the word translation, you usually talk about if you're translating English to French or something like this. You're switching from a nucleic acid to a polypeptide. So this is clearly a translation, to go from a nucleic acid to a polypeptide. So this is translation. This is transcription, the making of RNA. So hopefully that's clear. And so I put this picture on here because most living things on the earth are prokaryotic. And so transcription, the making of RNA, in this case messenger, happens right here in the cytoplasm, and then handy like this, the messenger RNA is translated into a protein also in the cytoplasm. If this was a eukaryote, I could do it. I can make it into a eukaryote. I would just go like this, like that, put the DNA in a nucleus. And then, as you would imagine, the RNA has to then leave through a nuclear pore. So translation is where proteins are made from RNA. So translation occurs on the ribosome. Transcription is the making of RNA. And so here's our eukaryotic cell. Notice much larger. Transcription is occurring in the nucleus, the making of RNA. And this is, I'll come back to this point at the very end. When you're making RNA, as it turns out, some of it needs to be spliced out or edited because it's a little long. And then that edited messenger RNA goes out and it makes a protein in the cytoplasm, or it's translated into a protein in the cytoplasm. So most of the transcription occurs in the nucleus and translation occurs on ribosomes. That's the point there. So, as I mentioned, even though RNA is produced in the nucleus, it also needs to be edited a little bit. It needs to be modified, called RNA processing, before it's actually exported out of the nucleus. And so let's cut right to the most important detail. So we're going to look at a closer look of transcription. And one of the big details is, you're like, wow, detail, do we really need it? Yes, I believe we do. So DNA makes RNA. We're like, okay, that's very good. That's what transcription is. But perhaps to speed this up, we might need an enzyme to do that. And you guessed it, we do. And so what would be the name of an enzyme that could speed up the making of RNA, if you can think of it right now? In other words, to catalyze the polymerization of a ribonucleic acid. You might guess it to be called RNA polymerase. and it's one of the key enzymes of all in biology. RNA polymerase, because it literally copies the DNA into RNA, and that's what we're going to look at and how RNA polymerase works. And so here's a picture of RNA polymerase shown here in blue. Redraw it like this. And there's its active site, and it's literally attaching onto DNA and opening it up. And so that's pretty cool right there. It's a real picture, computer enhanced picture. And so messenger RNA is transcribed from a template of DNA. In other words, sort of the master nitrogen base sequence. And the RNA polymerase can do sort of the work of two enzymes. You may be familiar with DNA replication when we talked about DNA helicase breaks the hydrogen bonds and unwinds the DNA helix. In this case, RNA polymerase can do that. It attaches onto DNA, and when it attaches, it opens it up, and it can also add new RNA nucleotides to the DNA. And so let's talk about that. Here's another picture of RNA polymerase. And so let's do this. Let me go this, and I'll add the double helix of DNA like this. And so what it does is that it can open up the molecule like this. Here's DNA in red. Let's do this, DNA. And so do you notice when DNA opens up, it's going to have exposed nitrogen bases like this on both sides, like this. And so what RNA polymerase does is it looks around, now we're in the nucleus, it looks around in the nucleus for RNA nucleotides. And if you remember, RNA nucleotides, I'm going to make them, let's say, blue. So RNA nucleotides are going to be blue. They're very similar to the DNA ones, but they're a little different. They have the ribose sugar and the base uracil. And so what's going to happen is there's going to be a uracil, a adenine, a cytosine, a guanine. And it's like, where do these RNA nucleotides come from? They also come from our food. We eat these things. And so these things are floating around in the nucleus. And so RNA polymerase comes along and it looks at the sequence. I'm just going to make up this particular sequence of this gene. I'll say it's an A, T, T, C, C, G, T, A, A. And so obviously that would be complementary down below. So what RNA polymerase does is it brings in these new RNA nucleotides. So A, if you remember, bonds with thymine in DNA, but since this is RNA, it's going to bond with a U, and then this is going to be an A, and then an A. You see how this is working? And a G. This is what the enzyme is doing, is what I'm doing, is bringing in free-floating RNA nucleotides and polymerizing them together. T binds with A is going to bind with U. It goes like this. So this is RNA. This is how RNA is made. Notice how it's single-stranded, but it's made from the double-stranded DNA. So this is called elongation, where it's elongating the RNA. Or in other words, it's transcribing it, making it bigger. And so if you think about it, elongation is important. That's right here, the RNA coding sequence of the gene. But the question that you could ask before that is, geez, there's so many different genes. How does it work? How does RNA know where the genes are? Does it just start grabbing a hold of DNA and start just transcribing it? And the answer to that is there's a particular area where it attaches. So this is kind of like your start area. I can simplify this and say that this is an area called start, and this is an area called stop down here, and then this is where you're making your RNA. But specifically, they're called the promoter sequence. So it's a series of nitrogen bases in DNA where RNA polymerase attaches. So let me animate that. RNA polymerase attaches here at the promoter, and then it moves along like here. And as it moves along, it'll start making the RNA. And then when it reaches this terminator sequence, that means the transcription is finished and the RNA is done. Here's your RNA. And then the RNA polymerase falls off. So this is where it attaches, and this is where it falls off. Hopefully that was clear. Let's actually, to make it really clear, let me show you this little video clip of that process right here. Let me go over here and show you that. I think this is pretty good. And so here you can see inside the nucleus, this is where transcription is taking place. And so as I mentioned, here is this promoter area. This promoter sequence is where the RNA polymerase, shown in orange, is going to attach. And then it moves along, it opens the DNA up, and so it exposes one of the strands for which it's going to copy, called the template strand. And then these little pink small nucleotides are RNA nucleotides. And it's going to elongate that, and it's going to continue on, as we saw earlier, and it's going to make many RNA molecules. So it's polymerizing right here, so there's the RNA. All the RNAs are made the same way. They're being transcribed from the DNA. And then it moves along, until, if you look at a close-up here, I really like this. This is showing how RNA polymerase is bringing in new nucleotides. And notice how U is with A, U is with A. And so here's a DNA with a G. Which one would be coming next? Would be a C, and then this one would be a G, and this one would be an A. Let's take a look. And so yeah, there's an RNA, there's another G, there's an RNA A. It moves along, and so this is your pink RNA right here. So that can be any of the types of RNA, and then it'll move along until it actually reaches the terminator, and that means that transcription is completed. And let's say that this is messenger RNA, and then it would then need to be edited, and it would be then good to go and leave the nucleus. So hopefully that was a good animation. I kind of like it. And so let's go back here to the notes and see how we can do this. And so again, promoter is where RNA polymerase attaches, the gene is where it's transcribed, and terminator is where it finishes. And so here's the same diagram, a little bit more detail. So here's the promoter sequence, which is a sequence of nitrogen bases. This white cloud is RNA polymerase. So this is called initiation, where it attaches. There's a little bit of complexity to this. We won't go into it in this level of biology, but it's pretty cool. RNA polymerase itself can't bind to the promoter. It actually needs a mediator protein and these other proteins called transcription factors. It's pretty neat. And even these other proteins called activators. And so all this attaches to the promoter, and then RNA polymerase is good to go. But nevertheless, it's the same point. RNA polymerase attaches, it starts to transcribe. Here it is making RNA, and so that's the elongation process. Right there you can see RNA is being produced, so it's initiation. Elongation is how you're making RNA, and then termination is when you reach this sort of red area right here where termination is complete, and that means RNA polymerase jumps ship. And so elongation is pretty neat. It happens really quickly. I don't know if you can get a sense of this, but the way I animate it, it sort of like brings one in really slowly, but it's approximately 60 nucleotides a second, so 60, 60, 60. It's really quick. And so this enzyme is pretty busy bringing in RNA nucleotides. And this is how RNA is made. The polymerase moves along, unwinds the helix, and adds new RNA. And so I actually do have a little animation giving you just a small sample of how fast this process is working. I find this is kind of neat. Check this out. So there it is. Here's the initiation complex. There's RNA polymerase in blue. DNA is in red, and that yellow streaking off the back is RNA. So this is RNA, and these little things flying around are RNA nucleotides. So there's the enzyme in blue. So look how fast it can make RNA. So it's cruising along. So it's reading the gene. And say this part right here is RNA, right there. And so you might say to yourself, well, what's going to happen once RNA is finished being produced? You're going to reach a termination sequence. And when you reach a termination sequence, you guessed it, RNA is finished, RNA polymerase falls off, and then the RNA leaves the nucleus. So that's kind of that's kind of a crazy video, but I like it. And let's go back up here. And so elongation, it polymerizes the RNA. Here's another picture of that, of elongation. Here's RNA polymerase. Now you can get a sense how fast it moves. So it's cruising. There's that big strand falling off of it of RNA. Could be messenger RNA. And then it reaches a termination sequence or terminator sequence, and that means that it's finished. And so I mentioned to you before that before RNA leaves the nucleus, it needs to be edited, and that's for this reason. DNA, let me extend it, is a really, really, really big molecule, and as it turns out, I mentioned the segments of DNA are called genes, and those are what code for the proteins. As it turns out, the genes can also be called exons. In other words, these are the regions of DNA that are actually going to be expressed into a protein. And so it's sort of like, if I can use this analogy, it's sort of like if you're filming something at home with your own digital camera, and you're filming, and you're filming for about 5 minutes, but in fact, not all of it you really want it. And so you can edit it down to about 30 seconds. Maybe that's how these videos should be handled. But there's regions that are really important called exons, and then there's regions of DNA, we go green here, which are non-coding. They're kind of unknown in their origin, but they're not important in terms of making a protein, and those areas are called introns. And so those are regions of DNA that do not code for proteins. So you've got good exons, this is the good stuff, and then you got this sort of stuff that needs to be edited, stuff that needs to be edited, and good. So you have good, good, and good. So let me label those. This is good, and this is good. So you want the exons. And so I mentioned before that there is this other type of RNA called small nuclear RNA. As it turns out that once messenger RNA is produced, it's produced both good, and bad. And the small nuclear RNA comes along and it edits out the introns. And so this small nuclear RNA attaches with other proteins, I won't go into detail, but I could, called a spliceosome, and it removes the introns. And so the good exons, A, B, and C, are then flanked side by side like this. And so now this is a good messenger RNA, and it's nice and small, and it leaves. The truth is, before messenger RNA leaves, actually on one side of it, on the lead side, which is something called the 5', a little cap is placed on it. It's a guanine with a methyl group, and that 5' cap is very important and helps it attach to the ribosome, and it actually helps it to leave the nucleus, and it also helps it to live a little bit longer in the cytoplasm. And then before the messenger RNA leaves, a big adenine tail is attached, and we believe that that is important too, to let RNA live a little bit longer in the cytoplasm. And so take a look at this. This is going to be our last little video clip of the process. And so I really like this one. Let me see if I can make it a little bit larger. We're in the nucleus. You can notice that some of the DNA is wrapped around histone proteins, and this little orange guy is RNA polymerase. And so what's going to happen is RNA polymerase moves along, and it attaches to the initiation, the area called the promoter. Then it's moving along, adding RNA nucleotides. You'll notice here the RNA is in red. And then it falls off, but before RNA is able to leave the nucleus, it also needs to be processed. And so a little 5' cap is added, a poly A tail is going to be added at the very end over there, and then here's the small nuclear RNA, which is removing the introns. There's the adenines attaching, and so here's your completed messenger RNA, and it's going to be leaving the nucleus like that. So that's pretty neat. And you're like, well, where's it going? That's a good question. Where is it going? the answer to that question is our next conversation. Our next conversation is going to be about translation. So once messenger RNA leaves the nucleus, it's going to go out to the ribosome. And the ribosome is going to be where proteins are made or translated. And that's going to involve all the RNAs getting together, messenger, transfer, and ribosomal. So I hope you enjoyed that conversation on transcription. Thanks for watching.